bacteria have a lot of different options in the way they can metabolize a variety of different molecules to release the energy that is stored within those molecules so that they can change that energy into a form that they can use in the form of ATP. We are going to approach Chapter 7 and look really in depth in one of these different pathways. We are going to look at how we break down, how we catabolize glucose in an aerobic environment. So we're going to look at glucose catabolism and all of the different steps breaking that down, ultimately utilizing oxygen as the very last step. We are going to look at this one pathway in detail with the idea that if you can learn this one pathway, we can talk about some different changes that take place, and we can get some really good examples of other different metabolic pathways using this particular one as our basis, as kind of a jumping off point. Before we get going today, I would actually like to turn your attention to some metabolic pathway cards that I have posted on Canvas for you to study from. This is just a Word document. It's meant for you to be able to print these off, cut the cards out, and then shuffle them up so that you can order these steps of molecules that will be formed during our different pathways that we're going to talk about. I'm really, with this particular chapter, going to pick a few important things that I want you to learn, and then we're going to skip over some of the other things that are just not as important to us because we just can't cover it all. This will help you understand the level that I want you to be familiar with these pathways. We do have an inquisitive that is going to cover more than what is covered here. And for this particular chapter, there are going to be some questions where you need to lower your confidence and just guess, and that's okay. But there are some really great questions in inquisitive that will help you understand and learn these different pathways and the different molecules that are made throughout this process. So for this particular chapter, I have no reading that's assigned for it, but do review the metabolic pathway cards that I've provided with you, and then go ahead, run through those inquisitives several times, and then there are a few questions that are assigned at the end of the chapter so that you can be familiar with this material at the depth that I want you to know it. All right, so without further ado, let's start learning about catabolism. So catabolism is the breakdown of food molecules, specifically to obtain energy. We have previously addressed that all organisms need nitrogen, they need a carbon source, but they also need an energy source. And we said that that energy can come from lots of different ways. We can get it through photosynthesis, we can break down inorganic molecules. We are going to be looking today, though, at glucose catabolism. This is the breakdown of an organic sugar, a monosaccharide. And we are going to see that there are a specific set of steps that are going to occur in order for that glucose to be broken down and release the energy that's stored within it. That energy will ultimately go on to make ATP, that energy molecule that we've talked about before. So when we look at catabolism, whether we're dealing with glucose or some other type of inorganic compound, we have to look at different, we have to realize that different species can metabolize different substances. And what determines whether or not an organism can break down a specific molecule really comes down to the enzymes that they have, which is determined by the genes that they carry. So their genes that they have will be expressed and used to make these protein enzymes. And then those enzymes will go to work carrying out chemical reactions. We have to consider that the enzymes that are present are going to dictate what is available for that species to break down. Additionally, today we're going to look at glucose and how it's broken down throughout multiple different pathways. But there are lots of different foods, essentially, that a microbe can use as an energy source, and they can enter at different points in this pathway. We're going to look at the whole long process, but realize that lipids and amino acids can kind of jump in here so that they can be broken down as well. We are going to be addressing some different energy carrying molecules today. In this particular section of your book, it mentions ATP, which we've talked about before. ATP stands for adenosine triphosphate. And in order to form ATP, we have to have ADP. And we are going to add on that additional phosphate and bring that ATP to its most energized form. We can utilize that phosphate to go and do work. And it can then be returned to its low energy state, ADP. So we go back and forth, back and forth. There's another couple of energy carrying molecules that you need to be aware of. One of those is NAD, which in its energized form is NADH. There's a different way in which we carry energy in this particular molecule. So here it's not a phosphate that is going to provide the energy for us. It's actually electrons. There will be 2 electrons that NAD will pick up, and it will be energized to NADH. There's also another one that we will address later. Your textbook doesn't talk about it at this point, but NADPH is another one that is an electron carrier. So electron carriers, we are just going to use their acronym. I don't really care that you know that it's nicotinamide adenine dinucleotide. It's a whole mouthful. And its name doesn't give you a clue as to its structure. So we're just going to shorten it from here on out. We're going to call it NAD, which is the low energy form, and NADH, which is the high energy form that has those electrons. But these electron carriers have a lot more energy than ATP. So we're going to see that as we break down glucose, we are going to produce lots of electron carriers. Ultimately, those will be used to generate lots and lots of ATP. ATP can be made in one of 2 ways. The first is what is called substrate level phosphorylation, which is where we have an enzyme that sticks a phosphoryl group on to form that ATP. We can also have oxidative phosphorylation, where we utilize the energy in our electron carriers to create an electrochemical gradient. That electrochemical gradient will be used by an enzyme embedded within the microbial membrane called ATP synthase. And its job is to take a phosphate group and tack it on to ADP, forming ATP. We're going to see that substrate level phosphorylation produces a little bit of ATP, which is cool. But when we want to produce lots and lots of ATP, we are going to create this electron gradient. And we are going to utilize oxidative phosphorylation to make lots and lots of ATP. Okay, so those terms are going to make a little more sense to you as we go along, but I want you to understand that as we go and I kind of throw those words at you, that's what they mean. It's just different ways of making ATP. All right, so we have to clarify a couple of different terms, and I always give the students the diagram that's on there that I've pulled from Google. So Leo the lion says GER. This set of acronyms is to help you remember the difference between oxidation and reduction in chemical reactions. So Leo, L-E-O, helps you remember that a loss of electrons is oxidation. GER, what the lion says, stands for a gain of electrons is reduction. It's counterintuitive that gaining electrons, gaining anything is a reduction. So if you have to write that on a piece of paper as you are taking the quiz, or you jot it down on your exam when you take that, you have to remember that those two words referred to the loss of electrons, oxidation, and the gain of electrons, reduction. So when we start talking about these electron carriers, we're going to use NAD as our example. NAD will be reduced to NADH when it gains electrons. So when we have these electron carriers and they're in their reduced form, they have high energy, and they can take those electrons and essentially deliver them to a series of proteins within the plasma membrane of our bacterial cells. These series of proteins are called the electron transport system, and they are going to transport those electrons that they receive from our electron carriers. So we'll talk more about this idea at the very end of this chapter, but understand that we are going to reduce our electron carriers, and they will pick up in that reduction electrons to take them to someplace else. All right, so I said we're going to focus on glucose catabolism. We are going to take glucose, which is a 6-carbon molecule, C6H12O6. It's depicted up there on the right side of your screen. And we are going to take it through its initial pathway called glycolysis. Glycolysis is the first step in breaking down glucose. What we are ultimately going to do is take this 6-carbon molecule, glucose, and break it into 2 3-carbon molecules called pyruvate. So the steps that are depicted over here are, your book does a really great job of this. So we've got our glucose molecule here. And we, in order to begin glycolysis, actually have to invest ATP. We have to use energy to kick off this metabolic pathway that's ultimately going to make us more energy in the end. So those 2 ATPs are going to have their phosphates broken off. We are going to use the energy there. So we invest those 2 ATP, and 1 phosphate is going to go here, and we're going to have another one here. That is going to form another molecule called fructose 1,6-bisphosphate. And I will tell you that I will always use in in the quiz and on your exams, I am going to produce questions that have both parts. So you'll have like the big long name, and then you're going to have the shortened version here as well. So we start off with our 2 ATP, we pull out that phosphate, and we form fructose 1,6-bisphosphate. So this is the shortened version here, Fru 1,6P2. So you can learn whichever one is easier for you. We then take that big molecule and we begin our catabolism. We begin breaking it down through a series of sequential steps. We are going to take this fructose 1,6-bisphosphate and we are going to break it down into 2 G3Ps, glyceraldehyde 3-phosphate. So, well, here's one, here's the other. They originated up here. So we're following those guys. Now, we're going to have those G3Ps are going to have electrons pulled from them, okay? So they are going to be oxidized, and our NAD is going to be reduced to NADH as it picks up those electrons. We are going to add an inorganic phosphate group, which is this little yellow guy that you see here. So now we've got this one here. We're going to add another. Oh, look down here. 1,3 BPG, so 1,3-bisphosphoglycerate. We've got 2 of those. They've both been changed from the G3Ps that we had, and they each have 2 phosphate groups. Now we're going to take those phosphates from each of them, and we are going to perform substrate level phosphorylation. So we're going to take 2 phosphate phosphates here and 2 phosphates here, and we're going to stick them on to ADPs, and we are going to form 2 ATPs over here, 2 ATPs over here. And what is ultimately going to be left at the end of this process is 2 3-carbon molecules called pyruvate. So in glycolysis, we take glucose, we invest 2 ATP to kick off our process. We're going to get 4 ATP out of the process for a net gain of 2 ATP. Does that make sense? So you add 2 ATP at the beginning to start, you get 4 back at the very end, and you have ultimately gained 2 ATP, which we said substrate level phosphorylation doesn't produce a whole lot of ATP, but a little bit's cool, and we can use that. Additionally, what we have produced here are some electron carriers. Those NADH electron carriers are going to be used at a later time. So they're important, remember them, but we're going to kind of sidestep that electron carrier right now to follow where our pyruvate is going to go next, which pathway, what are we going to do with it? So we've gotten a little bit of energy out, but there's still a lot of energy within that pyruvate. So we are going to continue following a cellular respiration pathway, so an aerobic and oxygen requiring pathway. Glycolysis is the start of a lot of different pathways. So realize that from here we can branch out in a whole bunch of different ways. We're going to follow one pathway. We're going to see that in cellular respiration, our 2 3-carbon pyruvate molecules are going to move on to the tricarboxylic acid cycle, or the TCA cycle. This may also be referred to as the Krebs cycle, with a capital K for the individual who characterized it long time ago. It's also sometimes called the citric acid cycle. All of those refer to names of molecules that are produced throughout this cycle. So it is a cycle. What we end up with at these series after these series of steps is going to be utilized in the next round. So what we're going to see is that we go through a couple of what I like to call grooming steps. Pyruvate can't directly enter the TCA cycle. So we're going to change it just a little bit, and we're going to use what is called acetyl-CoA to kind of usher in the breakdown product of pyruvate. Here, this is another place where if we're not metabolizing glucose in its full long pathway form, we can sometimes see lipids and amino acids sort of slip in at this point, and they can be broken down through some of these steps as well. So, like I said, we're going to focus primarily on pyruvate. The overall goal of the TCA cycle is to generate lots of electron carriers. So we are going to have, for each pyruvate, we're going to have some NADH that will be produced. So NAD is going to be reduced to NADH. We also said that another electron carrier that wasn't mentioned in section 7.2 is mentioned here. So FAD will be reduced to FADH. So these are both electron carriers that are going to be in their high energy states because they're going to have electrons that they have picked up through a series of steps that has worked to break down pyruvate. One of the byproducts of the TCA cycle is carbon dioxide as well. So if carbon dioxide is produced by an organism, this is where it's going to be coming from. So keep in mind that we're going to talk about one turn of the cycle because pyruvate is going to go through each of these steps. But remember that our glucose catabolism produced 2 pyruvates. So for each for each glucose that we have, we're going to produce 2 pyruvates, so 2 turns of the TCA cycle. So let's look at this in depth. We have at the top, this is the portion that I like to call pyruvate grooming. I think it's kind of like pyruvate preparation. So pyruvate is a 3-carbon molecule, and our first step is going to be to remove one of those 3 carbons, and we are going to produce 1 molecule of carbon dioxide. In breaking down that pyruvate in that initial step and taking that carbon off, we are also going to reduce NAD. So as pyruvate is oxidized, we are going to reduce NAD to NADH. So we're going to produce some electron carriers. This is our first step. Now, since we've pulled off one of our carbons, well, we're left with a 2-carbon molecule. And so we have a coenzyme A come in and kind of pick it up. So the resulting molecule, when coenzyme A comes in and kind of ushers that 2-carbon molecule in, is called acetyl-CoA. So we form acetyl-CoA, which is a 2-carbon molecule, and it begins to enter our TCA cycle. Okay, so technically we haven't actually entered the cycle. Each of those 2 pyruvates that are produced in glycolysis catabolism are going to undergo those initial grooming steps. And then we get to enter the cycle portion, as we begin to break it down, releasing some of that energy. So the reason that we will sometimes see this called the citrate cycle is because the first molecule that is formed is citrate, which is a 3-acid molecule. So that's where the name tricarboxylic acid comes from. So 3 acids there. So if we're bringing in 2 carbons with our acetyl-CoA, well, we're going to join that 2-carbon up with a 4-carbon molecule that is produced as the last step in our cycle. Oxaloacetate is a 4-carbon molecule. So we join that 4-carbon oxaloacetate with our 2-carbon, and that acetyl-CoA kind of drops those 2 carbons off. You can kind of think of it as dropping them off. And so then we have this 6-carbon citrate that we are going to begin breaking down. We pull off 2 of those carbons to produce carbon dioxide. We are going to reduce NAD to NADH. We are going to have some substrate level phosphorylation of ADP. We'll produce a little bit of ATP, which is cool. But through that series of steps, we are going to be producing lots and lots of electron carriers in their energized, their oxidized form. So we will have, for each turn, 3 NADH, and we will also have FADH2 that is produced. So those electron carriers are going to be really important later, but we're going to sidestep them for a little bit. So I have a great animation that your textbook has put together that shows these different parts of the pathway. But I realize that this can be a little overwhelming. So if you need to, go back, slide your bar back on the viewing bar at the bottom and listen to that explanation again. You may need to listen to it a few times, and that's okay. But really make sure that you're getting a handle on what is produced when. All right, let's take a look at this animation real quick. So this is on Canvas. You can watch this as many times as you would like.
Glucose can be completely broken down to yield energy through a few consecutive metabolic pathways, beginning with glycolysis. In glycolysis, glucose, which has 6 carbons, is split into 2 molecules of pyruvate, which have 3 carbons each. Overall, a small amount of energy is released, some of which is captured by energy-carrying molecules, NADH and ATP. Here the reactions of glycolysis will be arranged vertically based on the energy content of the substrate molecules. Glucose contains more energy than the 2 pyruvate molecules combined, which is why energy-carrying molecules can be generated during glycolysis. Glycolysis occurs in 2 stages. Stage 1 actually consumes energy, 2 ATP molecules, in order to prepare glucose for a controlled breakdown that ultimately releases more energy than is invested here. In stage 1, 2 hydroxyl groups on glucose are replaced with phosphoryl groups from 2 ATP molecules, and the molecule is slightly rearranged. The resulting molecule is called fructose 1,6-bisphosphate. This sugar then splits into 2 3-carbon molecules of glyceraldehyde 3-phosphate, each with a single phosphoryl group. These substrate molecules are primed to start releasing energy. In stage 2 of glycolysis, each of the 2 G3P molecules loses 2 electrons from 2 hydrogen atoms, converting NAD+ to NADH. NADH is a reducing energy carrier. The oxidation of G3P releases enough energy to add inorganic phosphate to each molecule, forming 2 molecules of 1,3-bisphosphoglycerate. Subsequent reactions transfer both phosphoryl groups from each molecule to a total of 4 ADP molecules, making 4 ATP molecules. Notice that although 2 ATP molecules were consumed in stage 1, 4 ATP molecules are produced in stage 2, for a net generation of 2 ATP molecules from glycolysis. At the end of glycolysis, the remaining 3-carbon molecule, pyruvate, no longer carries phosphoryl groups, but still contains a significant amount of energy that can be harvested by further oxidation. Under aerobic conditions, pyruvate will be completely oxidized by 2 additional pathways. In the first, catalyzed by the pyruvate dehydrogenase complex, pyruvate is stripped of a carbon, in the form of carbon dioxide, oxidized by NAD+, forming the energy carrier NADH, and combined with a molecule called coenzyme A, forming acetyl-CoA. In the next pathway, called the TCA cycle, acetyl-CoA combines with a 4-carbon molecule called oxaloacetate to generate a 6-carbon molecule called citrate, or citric acid. Citrate is a tricarboxylate, a triple acid, and gives the tricarboxylic acid cycle its name. This pathway is also known as the citric acid cycle, or Krebs cycle. In the TCA cycle, the 2 remaining carbons are removed in the form of 2 carbon dioxide molecules. The energy released by the removal is captured in energy carriers, ATP, FADH2, and NADH. As a result of these reactions, the 4-carbon molecule, oxaloacetate, is reformed and can be used in another round of the TCA cycle. From these catabolic pathways, glycolysis, the oxidation of pyruvate, and the TCA cycle, a small number of ATP molecules are produced. However, NADH and FADH2 are also formed, and they will transfer their high-energy electrons to the electron transport system, or ETS. In the ETS, the energy is transformed, ultimately producing a large number of ATP molecules.
All right, so I don't expect you on the TCA cycle to know any of those intermediate molecules, but do realize that citrate is what you start with and oxaloacetate is what you end up with. All right, so up to this point, we have generated a little bit of ATP through substrate level phosphorylation, where we've had an enzyme that has taken a phosphoryl group and just stuck it on an ADP, forming ATP. It's not a super efficient way of making ATP. We get a little bit up till now, but not nearly as much as if we create that ATP using a different form of phosphorylation, of adding on our phosphate. So what we've done up till now is we've made this little bit of ATP, but more importantly, what we've done is we have generated a lot of electron carriers, and they're all in that they're all in that reduced state where they have those electrons that they have gained. And so we now are going to turn our attention to what can we do with those electrons. Well, in aerobic respiration, we can deliver those electrons to a series of proteins that are embedded within the bacterial cell membrane. Just a note, and I probably should have said this sooner, make sure that you understand that what we're talking about here is prokaryotic cells, bacterial cells. If you have learned about generation of ATP in any other biology class, you have likely learned about it in eukaryotic cells, so where they would be talking about ATP production in mitochondria. This does not take place in the mitochondria. Remember that bacterial cells are single-celled organisms, and so they have no organelles at all. So everything that we're talking about thus far has taken place inside the bacterial cell cytoplasm, the fluid portion. And now what we're going to do is turn our attention to the cell membrane, that phospholipid bilayer. Embedded within that phospholipid bilayer are a series of proteins that are going to all be kind of clustered right together. We are going to see a sort of stair-step chemical reaction where we pass electrons from one of those proteins to the next. What we are going to do is use the energy in those electrons to pump hydrogen ions from the inside of the cell to the immediate exterior of the cell. Well, those hydrogen ions want to come back inside. We don't let them just come back through the cell membrane. What we do is we say, okay, you can come back through, but you have to flow through a specific protein. That specific protein is called ATP synthase. ATP synthase is an enzyme that will phosphorylate ADP for us. So that's kind of the overall process that we're going to talk about. We are going to use our electrons to generate what is called a proton motive force. And we use this proton motive force for a whole bunch of different things in bacteria. But the context that we are going to look at it for is how we generate ATP by having these hydrogen ions flow through ATP synthase and ultimately result in the phosphorylation of ADP, yielding lots and lots and lots of ATP, way more than we could get simply by substrate level phosphorylation. So this is aerobic respiration. So it's going to require oxygen at the very last step. So in aerobic respiration, we're going to see that where'd my mouse go? Where is it? There it is. It takes a minute to show up for me. So here we're going to see in glycolysis in the TCA cycle, we have lots and lots of electron carriers produced. Awesome. They have tons of energy within those electrons. They essentially drop their electrons off. Those electrons are going to be passed to the first transmembrane protein in this electron transport chain. And then these molecules now in their oxidized state are going to be low energy, so they can go pick up more electrons. But now we're going to follow the electrons that we've delivered. So these are called, this particular protein here is called NADH dehydrogenase. So it is an enzyme, essentially. It's going to accept those electrons, and in doing so, it is going to obtain energy that it will use to pump hydrogen ions from the interior of the cell on up to the exterior. And then we're going to pass those electrons on down into a quinone pool. This is a series of steps, one and then the other and then another. And each of those, as we are moving those electrons, are going to pump more hydrogen ions outside the cell. So we're going to get lots of hydrogen ions outside the cell, and they're all going, hey, I want to come back in. So ultimately, what we're going to see is that at the end of our electron transport chain, we've got cytochrome BO oxidase, which is going to be our last one, and it will pass those electrons on to waiting oxygen. This is important because this is the key. If you don't have oxygen here to transfer your electrons to, that whole electron transport chain shuts down. So we need that oxygen there so that we can transfer those electrons and pick up some hydrogen to form water. So as long as we've got oxygen and we can have those electrons passed down, we are going to pump lots and lots of hydrogen ions outside of the cell. And we're talking just in the immediate environment right outside that cell membrane. Well, they're like, hey, I want to get back in. So what happens is that the cell says, hey, you know what, you can have them flow back in, but you have to go through ATP synthase. And so that's what happens. As those hydrogen ions flow down through ATP synthase, this particular complex will phosphorylate ADP. And look at how much ATP is produced through oxidative phosphorylation. We have 34 ATP down here. Well, you compare that to the 2 that we got at glycolysis, and we got a couple out of the TCA cycle, but that pales in comparison to the amount of ATP that we can have when we fully break down that glucose. So again, we're going to watch an animation from your textbook. They give you a little bit more detail, a little bit more detail here than what I expect you to know, but the animation is really great. So we'll take a look at this one.
Respiration is the overall process of catabolism, from the breakdown of a substrate, such as glucose, to the reduction of a terminal electron acceptor, such as oxygen. For each glucose molecule that is catabolized from glycolysis through the TCA cycle, 10 NADH and 2 FADH2 molecules are produced. Each of these molecules contains energy that originated from glucose. Electron transport systems present in the membrane transport the electrons to oxygen, forming water, and in the process convert the energy into electrochemical gradients across the membrane. As the electrons are transferred within the ETS pumps hydrogen ions, also called protons, across the bacterial cell membrane. As a result, a gradient forms across the membrane, with a higher proton concentration outside than inside. In this way, the energy of the electrons from glucose is transferred to the energy of a proton gradient. The hydrogen ions will tend to move down their concentration gradient back into the cell, and they do so through a complex called ATP synthase. This flow of hydrogen ions through ATP synthase drives the formation of ATP from ADP, an inorganic phosphate. The production of ATP using the hydrogen ion gradient from the electron transport system and the reduction of oxygen is called oxidative phosphorylation. How does the electron transport system operate? An electron transport system consists of a series of electron carriers that sequentially transfer electrons to the carrier with the next higher reduction potential, that is, to a stronger electron acceptor. Electron flow begins with an initial electron donor, such as NADH, from glucose catabolism. Ultimately, all electrons are transferred to the strongest electron acceptor, which in this example of aerobic respiration is molecular oxygen. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex called NADH dehydrogenase. NADH dehydrogenase then transfers the 2 electrons onto the second functional component, a quinone, indicated Q. The quinone also picks up 2 hydrogen ions from the cell to balance the charge, generating quinol, QH2. The redox step from NADH through NADH dehydrogenase to quinol yields enough energy to pump 4 hydrogen ions across the membrane. Quinones and quinols diffuse within the membrane. Each quinol reduced by NADH dehydrogenase can transfer its 2 electrons onto the next ETS complex, cytochrome BO oxidoreductase. As part of the electron transfer event, cytochrome BO oxidoreductase pumps more protons. This terminal oxidoreductase now transfers the 2 electrons onto molecular oxygen. Oxygen has the strongest affinity for electrons of any other component of the electron transport system. In order to become fully reduced, molecular oxygen requires the electrons from an additional NADH molecule. In total, for every 2 NADH molecules oxidized, 16 protons are pumped across the membrane. The final 2 electrons join with oxygen and with 4 protons from the cell, producing 2 molecules of water.
Okay, so again, a little bit at the end there is more detail than what I expect you to know. I mean, I don't expect you to keep count of all of the different electrons and hydrogens that you need, but understand that oxidative phosphorylation is going to generate so much more ATP than substrate level phosphorylation. Okay, so what we've talked about now takes place in aerobic respiration, where we break down glucose. We said that there are tons of different possibilities, though, in terms of ways that bacteria can generate energy, can generate ATP. So another one of those ways is anaerobic respiration. So we're going to take what we learned and we're going to kind of put a little spin on it so that you can understand what anaerobic respiration was. We've talked about this previously in some of the other chapters, where we have respiration without oxygen. So anaerobic means without oxygen. Really, when we start looking at the details of anaerobic respiration pathways, we can have a variety of different terminal electron acceptors. So that's the last step in that electron transport system, but it's not oxygen. So whatever that terminal electron acceptor is, it is anything but oxygen. So some common ones would be nitrate, iron is another possibility. Salmonella is a bacterial group that kind of do some tricky things within the human body. If we have a salmonella infection, they use a molecule called tetrathionate as their ultimate electron acceptor. But this is not something that is typically available within the anaerobic environment of our intestines. So salmonella has a little trick up their sleeve. What these cells do is they initiate our own immune responses, and then they turn it for their benefit. So our immune response begins the inflammatory process and begins to react to these foreign bacterial cells that shouldn't be there. Well, one of the things that the white blood cells will produce is tetrathionate that salmonella can then use. So it's just one example of some kind of tricky things that our anaerobic organisms can use, but there are anaerobic organisms that can be pathogenic to humans. So just something to keep in mind. Additionally, when we talked about our oxygen tolerance tubes, we introduced the idea of fermentation. So fermentation is another pathway. This is a pathway that does not require oxygen. So there's not going to be any oxygen that's consumed. This is an alternative pathway where we aren't going to use an electron transport system. Essentially what we do as we begin to break down any number of different sugars. Fermentation is one where there's a lot of different variables that I guess not variables, It's a lot of different enzymes that we can use to accomplish the same process. But ultimately, fermentation is where we take a substance and we break it down, and then we take the electrons that were on NAD and we restore them to pyruvate. So recall that when we looked at glycolysis, there's some similarities there. We had to invest 2 ATP, but we got 4 ATP out for a net gain of 2 ATP. Well, that's essentially kind of the strategy that we're using when we use fermentation. We're not going to produce a whole lot of fermentation via ATP. We're not going to produce a lot of ATP via fermentation. But there are a lot of different ways in which bacteria can ferment some different sugars. Oftentimes when we have an unknown species in a lab setting, what we will do is we will investigate whether or not they can ferment different types of sugar. Do they have enzymes that they can use to break down those sugars through fermentation? And so that's what's depicted over here on the side. So these media over here have a pH indicator in them. Well, fermentation produces acidic byproducts. And so what happens is if a species is inoculated in a media that contains a sugar they can ferment, in an anaerobic environment, they will break it down and produce these acidic byproducts. Well, those acidic byproducts will cause the media to change pH. And so you can use a pH indicator, detect the change in pH just by having your media change color. Additionally, you can determine whether or not fermentation pathway of your organism that you're growing produces carbon dioxide. So in each of these tubes down here, these are called phenol red broth tubes because of the pH indicator that's in them. But inside they have a little upside down test tube. And so if the bacteria that is growing ferments that bacteria and produces CO2, what you'll see is a little gas bubble. Oh, where did my mouse go? There it is. A little gas bubble that gets trapped at the top of that inverted tube. And so that gas bubble tells you, hey, this particular organism not only can grow, it can ferment that sugar that you've provided it, but it is a gas producer as well. So sometimes we can use these techniques to learn about the different pathways that an unknown microbe is using, and it can give us a clue as to what that microbe is. All right, so as far as trying to understand all of these, new crazy biochemical terms, I would like you to focus really on the level at what is covered in this PowerPoint. I've provided the metabolic pathway cards to help you study. Print those off, cut them out, shuffle them up. The name of each part of the pathway is at the top, and then the different biomolecule is there. Can you put them in order? There are a few objective review questions and review questions that you can study. Additionally, you've got the inquisitives. There's some phenomenal questions that are in there. Like I said, there's some that you're going to have to lower your confidence level and just guess on, and that's okay. But if you have questions about this or you're unclear on some of the different steps, please email me and let me know so that I can clarify some of those things for you.